Okay, so hello everyone. Thanks for being here for another fine seminar. I'm your host today. I'm Lauren Hayes. I'm at the University of Tennessee at Chattanooga, and I'm really happy to have everyone here and to join us in the seminar. Just a couple of brief announcements before we start. Next week, we have Allison Bell, who I believe is at the University of Illinois, will be presenting. So I hope that many people can make that. The seminar today will be standard, about 50 minutes, 45, 50 minutes in length, and it'll be followed by a discussion. And we ask that, during the discussion, please, send a question mark into the chat, and I will pass those questions on to the speaker so that we can have a nice discussion. And we're trying to build a community here. So when you ask questions, please tell us your name and where you're from. And if you're on YouTube, there's a bit of a delay, of course, but I am monitoring YouTube and will relay your questions to the speaker. So please post questions in the chat over there if you're interested. So this brings me to today's speaker. I'm really happy to have Clint Penick present today. Clint is an assistant professor at Kennesaw State, which is a university in I believe, the Georgia system in the United States. Clint got his BS degree from Florida State University and then a PhD at Arizona State. And he's held postdoc positions prior to his appointment at Kennesaw at places like NC State and the NC Museum of Natural Sciences, and also at Arizona State. I hope today you'll find that I've seen him give a seminar before. He gives a really fascinating talk. And his research is really interesting because I think it spans multiple disciplines. It's integrative, truly integrative. He thinks about evolutionary mechanism, evolution, ecological conditions affecting social insects. And then he also thinks about, physiology and how that relates to ecology, behavior, and evolution. And remarkably, his research has spanned from various ecosystems in the tropics and also in the streets of New York City, which is really fascinating. He's been really productive early in his career. I've counted over greater than 40 publications in great journals, including Proc B, Evolution, Ecology and Evolution, Animal Behavior, AmNat, Behavioral Ecology. And in fact, one of these papers, I was listening to the news or reading New York Times, and I heard his voice or read his statement. So he got some big press on some of this cool stuff that he's been cranking out. And indeed, he's really been promoting his research and therefore our field broadly through outlets like the BBC, National Geographic Channel, New York Times, Popular Science, and a number of other, including one that I found really interesting, The World of Cheese. I'm not quite sure what that was, but I saw that on his website. He's also been at Kennesaw. They have a strong teaching component, so he's trained a large number of undergrads and works with master's students as well, integrating them, or having them do projects. So he's been really sort of across the board a professor, which is really cool to see. And today he's going to be giving a seminar. And Clint, I apologize, I lost the title.
No worries. I have it on the first slide.
Embarrassed to say that, but go ahead, Clint. Please share your screen and we'll see his seminar. So thanks, Clint, for being here.
Yeah, thank you for having me. I'm really excited to present to you all today. Let me know, can you all see? I'm sharing my screen now. I think it should work, right? Yeah. And so this is an exciting opportunity for me because a lot of my research in the last 5 years or so has shifted more towards ecology. As Lauren mentioned, I do research in New York City on urban ecology and some work on climate change. But today I decided to take a step back and dig back into some of the work that I started as a PhD student up until my most recent paper, which came out this summer that Lauren had mentioned that was in the New York Times. And it's all on these ants here that I'm going to talk about today. So the title that I had submitted is Dominance Hierarchy Formation and Brain Changes in a Socially Flexible Ant. That title is pretty long and pretty specific. And so maybe a better title would have been Ant Game of Thrones. because what I'm going to be talking about is queen development and how a reproductive division of labor emerges within social insect colonies. And this is one of the defining features of eusociality, so organisms that are fully social, eusocial, in that there's some members of a colony that do all the reproduction, and then there's other members that give up reproduction entirely and just help raise the offspring of those reproductives in the colony. And so I'm going to be talking about this at a couple stages, but all focused on this ant here, which I've been using as a model since graduate school. It's called the Indian jumping ant, saltator, that's its scientific name. This ant has become fairly well known in recent years because it was the first ant to have its genome sequenced, first of two, so they were both published together in Science in 2010. Shortly thereafter, Roberto then published the first epigenome of an ant. So he mapped the methylome of this species. And they've been using this now as a model to try to understand epigenetic mechanisms of gene regulation. And then most recently in this thread, I collaborated with a group where we were able to develop the first mutant ant line using CRISPR-Cas9 with this species. It was also one of the first two, so there was another species that was published in the same issue of Cell at that time. So a lot of this ant has been used in this sense for a lot of high-level genomic work to understand genes underlying the evolution of sociality. And I'm not going to be talking about any of that today. So I've collaborated on a lot of those projects, but today I want to focus more on the natural species and the behaviors that lead to some of the social organization patterns that we see in this particular group. So the ant, as its name gives away, the Indian jumping ant, it's from India. And more specifically, they live in this region in the southwest coast, the Western Ghats. It's a tropical region. The site that we collected from and where I've worked is this picture on the right. It's called Jog Falls. I think it's the largest or the tallest falls in India. It's quite beautiful. And these ants, they live in the forest that surround these falls areas. So when I've worked with them in the past, I flew out, and the first time I actually went to work with them, my PhD advisor sent me alone. I had never seen these ants in the field before, though I'd worked with them in the lab. And it took me fully 2 days of searching these forests before I found a single colony. And I want to point that out because these are extremely rare species, especially now that we think of them as a model for studying the genomics of social behavior. In the wild, they're quite difficult to find. Even when I got really good at finding their colonies, I calculated that it took me on average about 8 hours of searching for every colony that I found. So they form relatively small colonies that live in the leaf litter. And then they have foragers that singly forage. So they leave the colony. They have really big eyes. They're visual hunters. This is a picture of one, or a video of one in the lab. So they'll leave the nest and they'll look for small insects like these crickets here. And then this one just did it. They jump. So that's the other half of their name. They're from India, but they can also jump. And this is a rare trait among ants. You can see this one here. So they pounce almost like a leopard, and she's got herself a cricket, and she'll paralyze that cricket and take it back to the nest. And this species, they only capture live prey. So they're not like ants that you might see that feed on carcasses or dead insects. These are this species only hunts live prey. And so while they're unique for being one of the few ants that has the ability to actually jump, what they're probably more interesting and it makes them more unique, and the reason why they become a model to study social evolution is that workers in this species have re-evolved the ability to mate and lay eggs. So all extant ants have lost this initially, or the ancestor to all extant ants initially lost the organ called the spermatheca that insects use to store sperm. So some ant workers can still lay eggs, but those eggs are unfertilized and can develop into males, but they can't start their own colonies on their own. But Harpegnathos and a handful of other this structure, and so they can actually mate. And then when they do that, they can then technically found colonies by themselves. So this is something, they've re-evolved the ability to be a queen, more or less. Harpegnathos has an interesting history that starts out more similar to that of a typical ant. So they do produce true queens. This is a picture of one here on the left. These arise at the larval stage, and they're born with, or when they eclose from the pupal stage, they have fully developed wings. They go on a mating flight, and after they mate with a male from another colony, they'll break their wings off and then found a colony, and they serve as the primary reproductive individual in that colony. So they lay all of the eggs, and then all of the workers then take care of those eggs. And this phase of the colony. In most ants, this is the entire lifespan of the colony. So the queen might live in this species, she can live anywhere from 1 to 5 years. After that, the queen goes through senescence and she dies. For a typical ant colony, that means the whole colony dies. So they're over at that point. But because in Harpegnathos, the workers can mate, what happens is that this triggers a dominance tournament. So when the queen dies, within a couple hours, the workers will sense it. They'll start fighting each other. These tournaments actually take up to 30 days. So they're very long tournaments to decide who becomes the next reproductive. And then at the end of these tournaments, what you end up with is, instead of a single individual that lays eggs in the colony, you have a group of individuals, and they're called gamergates. You'll hear me use that term. So we call this the gamergate phase of the colony. And so these are mated workers. In this case, instead of mating with males from other colonies, they actually mate with their brothers inside the nest. So they're highly inbred. We don't know how they're able to be so successful and be as inbred as they seem to do quite well with inbreeding in the lab. But then once they become dominant individuals, they express a pheromone that clearly identifies them as gamergates. All the other workers go back to just being regular workers. They take care of brood, and then the colony becomes stable. And then this gamergate phase can technically last indefinitely. In the field, it doesn't, their colonies seem to only last maybe 5 or 6 years before they die out. But in the lab, we keep these going perpetually. So a lot of the colonies I worked with had been collected in the early 2000s but we're able to continuously breed them in the lab. And then if you need more colonies, you can literally just split them in half. They'll fight a tournament for 30 more days, and then you'll have two more colonies. So they're a really nice lab model organism for studying social evolution. So today for my talk, what I'm going to be talking about is the origin of division of labor at both of these phases. So I'm going to start by talking about the origin of the queen caste and the development of morphologically distinct queens like this one here on the left. And then the second half of my talk, I'm going to focus to how these workers compete in tournaments. And so this is, a typical ant larva. In ants, larvae are typically totipotent in that they can develop either into a queen or worker, and it depends on the nutrition they get as a larva. So if they get better nutrition by some metric, they'll turn into a queen. If their nutrition isn't quite as good, they'll develop into a worker. And when I started working with this species, this is kind of the general model. There's some nutrition that goes into this ant. It causes a hormone cascade. Some genes are probably involved, though none of them have been identified in this species. And then at the end, they'll pupate as a queen or a worker, depending on what that food source is. And better nutrition can refer to two different things. So in honeybees, which are one of the best studied for queen development, workers or larvae that develop into queens are given a better quality food. So they're fed with royal jelly, which is a high proteinaceous compound that bees produce. And larvae that get royal jelly then can grow to a larger size, and then they pupate as a queen. And then workers can control this. So workers either feed a larva, they give it royal jelly, or they feed it worker jelly. In that case, they develop into a worker. In ants, there's not a ton of evidence that they're able to feed larvae different qualities of food, so that most ant species don't do something as like bees, where they secrete their own larval food like a worker jelly or royal jelly. Instead, how caste is determined in ants is thought to be up by the amount of food. So in ants, they've evolved this ability called trophallaxis, where workers can regurgitate food directly into the mouths of larvae. They can even request food back. So if they think they're overfeeding the larva, they can cause that larva to regurgitate and spit it back to the workers. And so in this way, ants are thought to be able to tightly regulate the amount of food that larvae get, so that larvae that eat more food become queens, and then larvae that are given less food become workers. However, there's one problem with this. And it's that trophallaxis is a more derived trait in ants. And so species like Harpegnathos actually don't have trophallaxis with larvae, and the early ant ancestors didn't as well. So, so it's unlikely that workers were able to use trophallaxis this way when queens first evolved in ants. So in general, Harpegnathos has a range of ancestral traits that make it more difficult for them to regulate the food that larvae get. So as I just mentioned, they lack trophallaxis. They're a predatory species, so they hunt live prey. They bring that paralyzed prey back to the colony, and the larvae feed independently on it. So there's no mediation. So basically what happens is they bring these crickets, in this case, into the colony, and then the larvae, they have long necks, and they start chewing on the crickets directly, and there's no interaction between the workers and the larvae at this feeding stage. To make this even more complicated in Harpegnathos is that the larvae are actually mobile. And so most ants, they kind of sit around and get carried around by workers, but here the larvae can scoot around. So I have a video of that I'll show. So this is a time-lapse video. I just fed this colony, so crickets are coming in from the top. And if you look at these two larvae here, you'll see in time-lapse, they kind of zoom in. They can sense that those crickets have been brought in, and they'll zoom in, and then they'll feed on them themselves. So all of these make it pretty difficult for a worker ant in one of these colonies to tightly regulate the amount of food a larva gets. So when I started working on them, I wanted to figure out, well, what might they be doing instead of controlling food? And to do that, I needed to figure out a way to get colonies to reliably produce queens in the lab when I wanted them to. So that started out by first just studying their development. And so they start as an egg, then they go through a series of 4 molts. These are called larval instars. So they go through 4 larval instars, and then after the 4th one, they pupate. And then whatever they are as a pupate at the end of the stage, that's what they are for the rest of their lives. So if they emerge as a queen, they stay a queen. If they emerge as a worker, they stay a worker. And then what I found is that if you apply this insect hormone, juvenile hormone, to 4th instar larvae, this will induce them to become queens. So this is a sensitive period in larval development. If you apply juvenile hormone to the 1st, 2nd, or 3rd instars, you don't get queen development. It only happens at this last stage of development. And what this did for me is that it gave me a tool to control whether or not colonies started producing queens, so I could study how they regulated it. So what I did is I set up an experiment where I took a group of colonies and I pulled out their 4th instar larvae and treated them with juvenile hormone. Another group that received a control, they were treated with hexane or acetone in this case, but it didn't have any juvenile hormone in it. And then I observed them over the next couple of days to see how the workers treated them, to see what they did differently. And this is a case where I was kind of forcing colonies to produce queens when they wouldn't want to produce queens. So my prediction here is that once these larvae start to develop as queens, the workers will try to do something to push them back to the worker pathway. And so this is an example of a control larva in one of these experiments. This worker is just basically doing simple grooming. Here is one of the treatment larvae. So this is a larva that received juvenile hormone. And what I noticed is that instead of controlling how they were feeding these larvae, the workers actually started to chew on them. And it looked like to me that they were stressing them out or doing something. And I thought maybe this had something to do with regulating queen development. And so what I did again is then I quantified the amount of this chewing behavior. We set up sort of a blind experiment, so I didn't know if the larvae had been treated or not. And then at the end, what I found was very clearly that when larvae are treated with juvenile hormone and are pushed down this queen pathway in colonies that otherwise didn't want to be producing queens, we saw a lot of biting, and then in control, they weren't doing this. Then we took some of these same larvae and then I put them with groups of workers that had either been taken from colonies that were in a position where they would rear queens if they had queen larvae, and then ones that displayed this biting behavior. And then what we found when we did this experiment is that in colonies where larvae that were treated with juvenile hormone received no biting, they produced queens. So there was a lot of queens on average produced in these colonies. In colonies where no biting was present, or where biting was present, however, still some queens snuck through, but for the most part, there was a reduction. And this is pretty interesting because this shows that it's not only queen development at this larval stage is not only tied to food and nutrition, but it can also be impacted by stress. And so colonies have multiple ways of regulating the development of their larvae. And we don't really know still how queens are induced in Harpegnathos, but we know that if they start producing queens and maybe in a season when they normally wouldn't produce queens, or when colonies are just starting out and they don't have the resources to produce queens, what they can do is chew on them and stress them out. And it doesn't kill the larvae, they still pupate as workers, but it prevents them producing queens that otherwise would fly away and try to start their own colonies. So once we had done this, we figured out the behavior that these colonies were using to regulate whether they produce queens or not, but we didn't know how they were doing it, how they were figuring out whether a larva was going to be a queen or not. So larvae, they don't look different. And this response where the workers start biting them, this happens within hours of treating them with juvenile hormone. So it's not immediate. If you treat them with juvenile hormone, you give them back to the colony, they treat them fairly normally, but about 12 hours later, it kicks in something, and then the workers clearly recognize these as developing as queens. And so what I thought, and sort of the logical guess here, is that there's some sort of pheromone that these larvae are producing when they go down a queen pathway. And so I conducted a literature search to try to figure out, what work had been done on larval pheromones in ants. And when I began this, the most recent paper back then even was from 1988. And it was a review that was titled with the question, do ant brood pheromones exist? And if you read the whole review, their conclusion at the end was no. That based on all the evidence that people had presented so far, there was no clear evidence of any ant brood pheromone even existing, let alone anyone ever having identified compounds that could be considered a brood pheromone. So knowing that this was going to be potentially more difficult than I originally thought, I decided to start with some easier experiments. And so what I did is every once in a while in our lab colonies, a colony would start to produce natural queen larvae. They could be recognized at sort of the end of their larval life because they were just really, really big. So I would take these larvae, and if you put them in a colony that's not raising queens, the workers go crazy. They do that chewing behavior. They bite them. They become very aggressive. And so what I did is that I just took a queen larva and I rubbed her on a worker larva from another colony to see if I could transfer any compounds just mechanically from one larva to another. And I did this with a worker larva as a control. So I rubbed a foreign larva on a colony's worker larvae to see if that would cause any effect. And then I rubbed a queen, and I rubbed either her head and neck region or the posterior region. And this is because I wanted to see if maybe there was a specific gland, like an anal gland, or a gland in the mouth, like a mandibular gland, where they were producing these pheromones. Or alternatively, I wanted to see if the pheromones were just generally distributed on the body. So if they respond to both the posterior and anterior regions, then it's likely that it's like in the waxy layer on the cuticle that covers the rest of the body. And so what I found is that with worker contact, unsurprisingly, I observed very little to no biting. But then if they've been rubbed on either the anterior or posterior of the queen, biting was very clear. So it was very clear that a signal was being transferred and that this signal wasn't located necessarily on a specific gland, but it, at least at this point, had coated the entire body. This was our, what we concluded from this experiment. So next, I wanted to look at what these looked like, what compounds were actually in these. And so this is a readout from a GCMS. And what you can see is that they look pretty similar. So most of the compounds are shared between these, but the queen destined larvae have a higher proportion of short chain alkanes in their profile. So they have more C23, C25, and less C29 in this case than worker destined larvae. And we tried different variations and mixtures of these compounds, and we know for sure that they're not responding to any one of these compounds on their own. But what I was able to do was extract larvae, queen larvae in hexane, which is a nonpolar solvent that pulls off primarily cuticular hydrocarbons that are on the outside of larvae. And then we extracted them from workers as a control and queens. And what we found is, like, sure enough, what you can, when you extract these nonpolar compounds, it causes biting when you transfer it to a larva. So in this case, we have a pretty good idea. We never got down to identifying the exact compound that would constitute a pheromone to identify a queen destined larvae in this case, but we did narrow it down that it's a nonpolar compound that's present generally on the surface. The thing I was most excited about this is I got to name it, and so I called it the Princess Pheromone. So this is the first example of, instead of a queen pheromone, these are future queens, so this is now called the Princess Pheromone. So to kind of wrap this section of my talk up, what we found is that the way caste is regulated at the larval stage is that it relies on increases in juvenile hormone. As juvenile hormone increases, it causes larvae to produce some sort of nonpolar compound hormone signal, and that workers in these colonies can perceive these signals and then make a decision whether or not the colony is producing queens or not producing queens. And if they're producing queens, then they won't do any biting, and they'll allow these larvae to develop as future queens. If they're in a position where they're not producing queens, bite them, use that to push them back down to the worker pathway. And currently, I think this is still probably the most complete picture of the regulation of queen development in any ant species that we have so far. And so I do just want to take a moment to point that out, that a lot of research on queen development has focused on the hormones within the larvae that result in queen development, and to a lesser extent, some of the genes that are involved in this. But this really requires selection both on the adult caretakers of the larvae in addition to the larvae themselves. And so to truly understand how these caste systems evolved in social species, I think it requires looking at the bigger picture and trying to connect a lot of these dots together. Okay. So that's the section on queen development. Now I'm going to switch gears and then talk about how, after this, the original queen of a colony dies, and then they go through this tournament phase, how a group of reproductives emerges and it's gamergates to produce a reproductive division of labor here. So this is a video of what happens a couple hours after you remove the queen from a colony. What you see is that workers start getting agitated, and they do this behavior here called antennal dueling. So it's a form of ritualized dominance. They're not inflicting any impact or damage on each other physically, but they just beat each other with their antennae. Another common behavior that you see during these tournaments is here, this one that's labeled in green. She's beating. So this is a clearly identifiable agonistic behavior where one individual bites another. And so there's a dominant individual and a subordinate here that's losing one of these tournaments. And by far in these colonies, it's this antennal dueling behavior that's the most common. And it's sort of an enigmatic behavior. And when we started this, we didn't exactly know what ants are gathering by, like, what information they're gathering or what signals they're sending to other individuals by doing this. So now I'm going to talk a little bit about how their hierarchy forms and some of the research we've done on that. So most typically, when you have dominance interactions in animal societies, you get something that's called a linear hierarchy. And I think probably most of you are familiar with these. You have an alpha individual and then a beta that's less dominant than the alpha, but then more dominant than the gamma, and so on down the line. And so you have this linear ranking where no individual at the top of the hierarchy is equal, but there's always one that's above it or one that's below it. There's a number of ant species where these linear hierarchies are found. Some other species kind of similar to Harpegnathos, where they've lost the queen caste and the workers have re-evolved the ability to mate. In most of these are so-called queenless ants because they have no queens in their life cycle, but they have ritualized dominance tournaments that are similar to what we see in Harpegnathos, and they produce linear hierarchies. Another form of a hierarchy that's present in animals is something called a despotic hierarchy. This term is sometimes used differently to refer to hierarchy systems in primates, but in ants, and I think there's some fish that display despotic hierarchies, In this case, what's different from a linear hierarchy is that there's one individual that's completely dominant, and then all the other individuals below them are equally subordinate. So there's no linear ranking system. There's just one dominant individual and the rest that are subordinate. And neither of these really describe what we have in Harpegnathos. And what we have in Harpegnathos is something that we call a shared dominance hierarchy. So we have a group of individuals that are all equally dominant. They all equally tolerate each other. There's no linear ranking system between them. And then kind of like a despotic hierarchy, all the other individuals are equally subordinate. And so this is something that's relatively unique to Harpegnathos. And when we looked at the literature to try to find other examples of this, the closest that we could find were examples of coalitions and alliances in higher vertebrates. So these are things where groups of individuals, like in dolphin pods, will get together and form a coalition. It's found in primates. There's examples of this in humans. And most of the literature about these types of associations focus on higher level cognitive processes, individual recognition among group members, and all of these sort of complicated behavioral programs needed in order to maintain and form coalitions and alliances. And none of those things are things that we expect ant brains to be capable of. So instead, we thought that this similar sort of, what looks like a similar dominant system in Harpegnathos should emerge through something like a mix of simple behaviors that don't require higher level cognitive processing. And so these are the three primary behaviors that are used when one of these tournaments starts. And so I'm just going to go through these individually. So biting is a typical dominance behavior where there's a clear winner and a loser. So what we think might be going on here is that there's a winner effect. The individual that's biting a subordinate might get a little boost here and raise their standing in the hierarchy, and then the one that's receiving the biting gets a loser effect, and so that they're a little bit lower after they get bit through one of these interactions. Another behavior that's also common in other ant species is called policing. Policing is sort of unique to ants in that instead of a dominant individual biting and attacking another potential dominant, policing is performed by a subordinate individual. And so a subordinate ant here will basically identify one of the ants that's competing in this tournament and say, you know, they look like they're not going to win, they're wasting our resources. And what they do is they bite and they hold on to them. And they hold on to them for hours at a time, sometimes up to 24 hours. And again, they don't kill them. They don't usually sting them. Sometimes it'll escalate to stinging, but very rarely do they do that. They just hold them. And at the end of that, usually their chances of becoming a dominant are zero. So once you get policed, you fully revert back to a subordinate worker. So this is sort of a neutral for the one performing it and a negative. And dueling is this behavior, again, that there's no clear winner or loser. And so in looking at this behavior, what we hypothesize is that instead of being one of these classic dominance behaviors where there's a winner and loser effect, we thought that it could potentially represent a winner-winner effect. So that two individuals that are dueling with each other are both getting sort of a medium boost and whatever sort of winner effect. And as they kind of move their way through the hierarchy until it converges around 30 days later on a group of camera gates. We have some physiological data to back this up. So I had done work on dopamine levels in the brain. Dopamine is one of the main regulators of ovarian activity in Harpegnathos. And workers, once they get policed, within hours, they show a severe drop in dopamine, sort of unsurprisingly. So they get policed and their dopamine drops, and then any sort of reproduction ceases after that point that they start doing. Also, of course, dopamine is part of the reward pathway. And so what we found is that when these tournaments begin, the individuals that are doing the most see an increase in dopamine. So there is some evidence that dopamine here might be mediating a winner-loser response that ties back directly to their reproductive status. But the way we really wanted to test this would have been to ask a colony, can you only do dueling or stop doing dueling or only do biting? But we obviously couldn't do that. So then I developed a collaboration with a behavioral modeler, Taka Sasaki, and we put together an agent-based model where we put these rules and then we studied how having these different behaviors in combinations affected the emergence of typical dominance hierarchies. And so here in this model, They all start out equal, and so they're all brown. Brown means they're equal. This little white square in the middle, that's the brood pile. And so the way that when these tournaments happen in a typical colony, the dominant individuals associate around the brood pile, so the larvae and pupae, and that's sort of the staging ground. So we put a brood pile in this model, which kind of helps the dominants interact with each other at a higher rate. So the higher level of dominance you have, in this case, we call it reproductive status or ovarian status. So the more closely you were to a reproductive, the more time you spent near the brood pile. As the model ran, in general, there was a sort of logistic increase in their reproductive status. So as long as they didn't interact with any other ant, their reproduction would go up. But when they bumped into another ant, then there was a decision tree that they could perform one of the three dominance behaviors that we had specified. So they could do dueling, biting, or a policing behavior could ensue. And then we programmed in the sort of winner-loser effects that we predicted resulted from those behaviors. So over time, what you start to see is that some of these members become gray. That means they become a full subordinate, and now they're just workers in the colony. And then by the end of our runs, what you would see is either a group of individuals that remain dominant, it's brown here, that are towards the form of the hierarchy, depending on what behaviors we put in here. And so what I'm going to show you here is ranking of their dominance. This is their reproductive status or social status on the x-axis. Members that are at 0 or 0.1 or 0, those are ones that have become fully subordinate, are workers. Ones that are at 1 are the primary dominant individuals in a colony. And so we ran this model where we only included biting, or then we included biting and added policing, or then we ran all three together to see what the hierarchies look like that emerged from this. And so when only biting was present, this is the sort of classic winner-loser response. What we saw is something resembling a linear hierarchy where you had typically one individual that would end up at the top, one that would be a beta, one a gamma, and so on down the line, and then a group of individuals that were also sort of subordinate at the bottom. When we added policing to this model, what it did is it pulled out all those individuals that were in between the dominant and subordinate. So when we added policing, we got a despotic hierarchy, basically. So you end up with one individual that's dominant and the rest are subordinate. What we were most excited to see is what happened when we added this winner-winner effect, which had never been examined before. And what we found is that when we added dueling and policing and biting together, we found something that resembled what we actually see the hierarchy look like in Harpegnathos. So this is an example of what we called a shared dominance hierarchy, where you have a group of individuals that emerge at the top, and then the rest of the individuals in the colony are all subordinates. So this is some support that we think we have the right idea here, or at least pretty close to what these behaviors or how they're functioning in the formation of these hierarchies. Of course, this is a model result, and to some degree you can argue you get out what you put into it. So then the next thing we did is do a literature review of all of the hierarchy types that are found in ants. And so there's a number of ants. All of these are queenless species where they've lost a queen and workers form some sort of dominance hierarchy. We classify these hierarchies based on the published accounts of their behavior. And they range, there's one species down there at the bottom, Diacama. They're the only one that has a true despotic hierarchy. And in that species, what happens is that whoever wins the tournament, they start out as linear, but whoever wins walks around and mutilates all of the losers. And the mutilated losers can never lay eggs again. And so it truly is that once they become fully established, there is only one, and then there's no contenders for that spot. Linear dominance hierarchies are common in a group of other species. And then shared hierarchies have evolved at least 2 times independent. Rhytidopona and Naptogenes are somewhat related to each other, so they potentially could have shared a common ancestor, but at least 2 times independently, these have evolved. And then we looked at all the behaviors associated with the formation of their hierarchies, and then noted whether or not dueling occurred. And what we found is that dueling did occur and only occurred in species that form shared hierarchies, and it's evolved at least independently in 2 of these groups, if not 3. And dueling is not present in these other species. And the other species that form linear hierarchies, they have a behavior that's similar to dueling, where they beat each other with their antennae, but it's not reciprocal. So what happens is it's more like a biting behavior where one individual, a dominant individual, beats a subordinate with their antennae that behavior, they call it antennal boxing. It's different than dueling, but dueling is only happens, we only classify something as dueling as if it's reciprocated back and forth, like what you would see in Harpegnathos. So based on this, we think that dueling is a pretty interesting, relatively unique dominance interaction that has this winner-winner effect, and it allows, simple animals like ants that don't have these sophisticated cognitive abilities to develop these shared dominance hierarchy structures that, at least on the surface, resemble some of the social systems that you see in higher vertebrates, but they get there using a different mechanism that doesn't require higher level cognition. And so the last thing I want to talk about today is what happens once one of these individuals wins one of these tournaments and they become a gamergate in the colony. So once they win, they begin laying eggs. The other thing they do is that they also display a fertility signal. So they have a cuticular hydrocarbon compound, sort of blend of compounds that identifies them as an egg. Is at the end of that 30 days, the members that win, the individuals that win those tournaments begin laying eggs. So they're already egg laying at that point, and they produce a fertility signal that's strong enough to shut down reproduction by all the subordinate workers. So this is sort of a key way these hierarchies are regulated. And once the tournament is over, then they respect the winners. So once this pheromone is in place, there's no more fighting. You don't observe a lot more fighting in colonies. They can stay stable for years at a time. And only once those gamergates die, or once they're removed, like we experimentally remove them sometimes, then that'll initiate another tournament. But if that doesn't happen, they're more or less stable. The other thing that happens is that their behaviors change when they switch. So one of the things is they no longer have to hunt for themselves anymore. So once they become a gamergate, they just live inside the nest. They never leave the nest again. And all their food is brought to them. And so they don't need their venom gland anymore. And so we see that their venom, they actually shut down venom production, and presumably they're taking all of their resources that would go into things like venom and other sort of body maintenance resources, and they're pushing them into their ovaries to produce as many eggs as possible. Another interesting thing that happens, and one of the reasons why people have been interested in the species as a genomic model, is that their lifespan dramatically increases. So a typical worker in a Harpegnathos colony lives about 6 months is the average lifespan of a worker. Once they become a gamergate, we've tracked them and they can live up to 5 years. And so this is potentially a 10 times increase in lifespan with no major morphological changes on the outside of their body, right? They have identical genetics. The only big difference is that they're laying a ton of eggs. Their behavior changes. They have some, epigenetic changes that are associated with it. But this is one of the kind of cool characteristics about Harpegnathos. And then the other big thing, and the thing that I got really interested in, was that their brain actually shrinks by 25%. Which is a huge change in brain volume. And the idea behind this, again, is similar to the venom gland. It's that brain tissue is metabolically expensive. It takes a lot of calories and resources to maintain an expensive brain. And so by shrinking their brain by 25%, these gamergates can then shift those resources into reproduction. And there's also, their life becomes a lot easier once they win one of these tournaments, right? So they don't leave the nest. They never see anything again. Presumably in the wild, they live underground, and so they'll live in total darkness. And so what we see is that the optic lobes are one of the primary things that shrinks in these. So they lose a lot of the mass in their optic lobes. They also don't have to make sort of the complex decisions that are associated with a forager. A forager has to leave the nest alone. It has to navigate like a complicated world of leaf litter. It has to find prey, capture that prey, and then remember how to get all the way back to its colony, which is pretty cognitively demanding, especially for an ant. But once you become a gamergate, you don't have to do that. And so we think that these cognitive demands are lessened. Some of the senses required for hunting, they no longer need. And so this is why you see this 25% decrease in brain size. And so all of this stuff, for the most part, I've been known when I was in graduate school working on these, but I was always interested in the back of my head, wondering what happens when one of these gamergates eventually loses their status. Can they go back to being a worker, or do they just die? And if so, if they are able to go back to a worker, how much of this is actually reversible? how much of this can they get back? And particularly the brain changes is what I was interested in, because I know like growing up, my mom always told me like, don't hold your breath too long, you'll kill your brain cells and you never get them back. I'm sure a lot of people heard that. The brain tissue is famously does not regrow after you lose it. It can rewire. There's a lot of neuroplasticity and a lot of research on that, but something like losing, a quarter of your brain, you don't think that you could get that back very easily. Long time, there's actually some research where this is actually true. So there had been research done on canaries that I had heard about in grad school, which made me start thinking about these questions, where male canaries have their brains actually shrink and re-expand seasonally along with the breeding season. And so they need a large sort of brain size to remember the complex songs that they must sing to attract female mates. But then outside of the breeding season, that excess brain is just costly. They don't get a lot out of maintaining it. And so they actually show these seasonal retraction in brain and then re-expansion. And this has been found in some other organisms as well. So there's a number of songbirds that do this. And I think there's some mammals that do this as well. But again, this is found primarily in relatively long-lived vertebrate species, higher vertebrates, and not in insects. So in insects, they have such short lifespans. Again, typically it's around 6 months for an ant. And so the ability to maintain reversible plasticity and brain size, people have thought there might not be any selection for that to happen. And people have looked. So so honeybees exhibit plasticity in their brain size associated with task performance, and you can revert them back to earlier to earlier roles in the colony. And they show plasticity in a whole range of traits, including their learning ability and lifespan, but their brain doesn't change. So that was one of the things that stood out, is that when you revert honeybees back to like a nurse status, their brain doesn't change back to the nurse brain size. It stays the size it was. People have also looked at this in Drosophila, and they found some regions of the brain are plastic, but nothing on the scale of like a 25% change in brain tissue that's plastic. So when we started this, we thought maybe this wouldn't work, but let's see what happens. And one of our big challenges in doing this work is that we had to think, how do we take one of these gamergates and actually revert it back to a worker without it dying? We had never tried to do this before. This happens naturally sometimes, but you might have to wait 5 years before a colony does this, and you have to check them every day. So what I ended up deciding to try, and what worked, is that I took gamergates, I identified them in the colony. So I would identify a pair. So one would be the control, I'd leave her in the colony. Another one I'd take out, and we put her in a box by herself. We gave her all the food that she needed. We took care of her, so she had everything she needed. She was just isolated from the colony, put in social isolation. Then after 4 weeks, we returned her back to the colony. And our idea was that just from the social isolation, we thought that might reduce her reproductive output to some degree, and that her fertility signal would reduce to a degree that she would no longer be recognized as a gamergate, and she would get policed and then reverted back to a worker. And so sure enough, 4 weeks was long enough, and that's exactly what happened. This is a picture of one of those workers. So this one in the center of the screen here, she has paint on her. She's painted orange and white. This is one of these gamergates we returned, and the response was quite extreme. So this is one of the most extreme examples of policing. Usually it's 1 or 2 individuals that grab and hold onto them. In this case, there was multiple individuals, and they grab her and they hold her. But like I say, they don't necessarily damage them in policing. They just hold them and immobilize them for up to 24 hours. And then after that, what we did is we tracked their behavior to see whether they did truly revert back to a worker. And so the first thing we looked at was just where they are in the nest. Gamergates, like I said, they always stay near the brood pile, and we have sort of an inside nest area and then a foraging arena outside of that. Gamergates are basically never found in the foraging arena, but after we reverted these gamergates back, they were found predominantly in the foraging arena. The control gamergates, again, were only found inside the nest. There were other changes in behavior we tracked. So as I mentioned, that gamergates don't hunt. Once they become a gamergate, they don't hunt crickets anymore. They actually lose their venom. And so we isolated control and reverted gamergates with a cricket, and we quantified whether or not they stung those crickets. Control gamergates never did. They would just stay in the jar with a cricket and let the cricket walk on their face and everything else. They would never bite them. They would never attack. But these reverted gamergates, within a couple weeks of being returned to their colony, already fully switched back to being hunters and performing that task. And then also gamergates aren't involved in nest defense. So if you open up a nest and kind of reach forceps in to pick up an ant, if you do that to a worker, a subordinate worker, they'll run out with their mandibles open and they'll try to sting you and everything else. But a gamergate will like duck and try to run and hide. And so we saw here again that their propensity to bite forceps and display colony defense behavior was totally returned in reverted gamergates compared to control gamergates. So we let them stay in these colonies. Like I said, that they have plasticity and lifespan. So what we found is that once they reverted back to workers, their lifespan also dropped back down. So they would usually live not more than maybe 2 months after we return them back. So generally around 50 or 60 days, we would take them out of the colony and sacrifice them to take a series of measurements on their bodies. One thing we looked at was the number of yolky oocytes, so the number of eggs they were producing, just to show that they had indeed shut down reproduction. And so you can see control gamergates are on the left here. They're highly reproductive. They typically have 6 to 7 oocytes. Reverted gamergates had virtually 0. Every once in a while, they might have had a developing oocyte, but virtually 0. They were more similar to foragers. And we look at foragers and inside workers here, which are both subordinates, but an inside worker is more of a nurse that takes care of brood, and a forager is the individuals that are older that leave the nest and do hunting behaviors. So we knew there was a change in reproduction. The next question was, do the brains change? And so this is the result from the brains. There's a lot more overlap than maybe we would have predicted, but what we did find is that we found that yes, like indeed that compared to foragers, control gamergates did show around a 20 to 25% decrease in brain volume. This is total brain volume, and reverted gamergates got that back. So reverted gamergates became, they were no different statistically from foragers in their brain size. So they reverted back to a typical forager brain. One of the things that previous studies hadn't looked at was these young workers, inside workers. And young worker brains are more similar in size to control gamergates, but there are some differences. So there is still evidence that once an individual becomes a gamergate, they are displaying a reduction in brain mass, a true reduction, and that they're not just staying at the inside worker side. And one way we were able to tease that part was by looking at the different regions of the insect brain. So insect brains have 3 major regions. They have the central brain, which is coded here in yellow. This is the region that has higher level processing. It has this region called the mushroom bodies, where decision making and different things like that happen. They have the optic lobes on either side here. These are just behind the eyes, and those are in visual processing. And then they have the antennal lobes, which are the ones in pink shown in the photo. And those are where they process chemical and pheromone information. So one of the things that we found is that the biggest changes that we saw were in the central brain. So the central brain is the main thing that's contracting and expanding when individuals become a gamergate or lose that status. We found no difference in the antennal lobe, so the antennal lobe volume stays the same across all individuals. And the optic lobe volume is the one that showed a difference pretty clearly. So even if an inside worker wins one of these tournaments, becomes a gamergate, it's still clear that they're losing mass inside the optic lobes. So this is pretty clear here. And the optic lobe was the thing that showed the least change, so in reverted gamergates. So it lagged a little bit behind the central brain in re-expansion. But so all of this together showed us that, all of these systems in these ants that allow them to perform various roles as workers or as reproductives are plastic, and they remain plastic for their entire lives. Which is pretty interesting, and it separates it from what happens at the larval stage. Once a larva becomes a queen, there's no going back. And so just to kind of summarize some of the main points throughout the talk is that sort of the evolution of a reproductive division of labor in both larvae and adults relies on plasticity and behavior and physiology together. In larvae, this means that there's been selection on the behavior of adults that take care of larvae in addition to the larvae. So if you're looking for a key gene that creates queen development, you can't just look at larvae. You also have to look at the genes that are controlling the behavior and the responses of the adults that take care of them. And that's something that's been neglected, at least in a lot of the literature and the research focused on understanding genes underlying sociality. And then finally, in adults, plasticity remains reversible throughout their entire life. And this was sort of surprising to us. We don't fully, we have some ideas about why this might be true, but there is some cost. There are some costs to plasticity, and there are questions about why an individual that becomes a gamergate would still maintain whatever's needed to revert their phenotype back to the worker stage. I'm happy to talk about that more later, but in any case, what's interesting at this stage is that they remain reversibly plastic. So with that, I'd like to thank collaborators on these projects, undergrads. Some of the photos came from these people, and the funding sources primarily came from HHMI for this work. And I'll take any questions. Thank you all.
Well, thanks, Clint, for a fascinating talk. Clint, if you could stop sharing your screen just for a moment. Thank you.
Right.
So now we have time for questions. Really cool system, really cool talk. And at this point, if people would please put question marks into the chat, and I will relay those questions to Clint. Also, if you're on YouTube, please put your questions there, and I will relay them as well. And remember, when you ask a question, we ask, again, we're trying to build a community here. So please, just state your name and where you're from, so we can get to know each other a little better. All right, so the first question we have comes from Eduardo. So Eduardo, please.
Thank you, Clint, for a very, very interesting, engaging presentation. It's really, like Lauren was just saying, a fascinating system. But by the way, I'm Eduardo Fernandez Duque at Yale University. And I have many questions. I'll start with a very general one that speaks to what I think it's a common interest in natural history. So you were telling us how this is a rare species and species. So how did your advisor, or why did you guys get started with this rare ant species that suddenly has become this fascinating new system?
Yeah, so it started really a discovery of an ant researcher named Christian Peters, who recently passed away this year. But he's the person who originally is kind of he was a really cool scientist that went around and looked for weird ants. That's what I would describe him as, and he was very, very good at finding them. And so he became interested in queenless ant species, where you dig these colonies up, there's no queen, and then he was one of the early people to start dissecting them and finding out, well, even though there's no queens, sometimes he usually found like one worker in a colony that was filled with ovaries and was laying eggs. And so he's the first person. He just happened to be at Jog Falls, which is one of the reasons why we work there, and he found colonies and started digging them up. Noticed that some of the colonies had queens and some of them didn't. And when they didn't have queens, he would do a dissection of literally the whole colony. He'd dissect them, and then found that one of the unique things about Harpegnathos is that they have a group. So that's one of the things that there's only, like I said, 2 other genera that show this same pattern, where they've evolved queenlessness, but they also have a group of workers that do reproduction. And so he was working with Bert Holdovler at the time, who was also he's one of the co-authors with E.O. Wilson on the ants. And so he shipped all these samples back to Bert, and they published the first paper describing sort of this really, at the time, I think that it was first discovered in the 1990s. And at the time, they were really, really different ant species. Now that there's more that have been discovered like that, but there's still, there's 15,000 ant species that have been described. There's probably dozens that show behaviors something like this. So it's not a lot.
Thank you.
Okay, so Carsten is next.
Yes, hello, my name is Carsten Schradin. I'm a researcher at the CNRS in Strasbourg, France, studying mammalian social evolution. Thank you very much for really a fascinating talk, opening an entire new cosmos for me about these ants and gamma gates. And as such, I also have to apologize for my question, which you might have answered during the talk, but as there was so much new information for me, I will still want to ask it again. These gamma gates, of course, are very interesting. Did you say how many there are per colony and how it is decided, whether it will be 1, 2, 4, 5, or whatsoever, the number of gamma gates emerging from these tournaments?
Yeah, so it's a really good question. I didn't mention that, but there's always a group. So it's never that one wins. And typically it's tied to colony size. So bigger colonies, if they start out bigger when one of the tournaments starts, they typically have a higher number of gamma gates. And the proportion, I think, is somewhere between 5 to 10% of the colony initially. And also, the interesting thing about this is when the tournaments start, virtually 100% of workers participate in the tournament. So even some of the older workers will start in the first couple of hours doing some of the sentinel dueling, but they usually get pushed out pretty quick. And yeah, there's other interesting questions about this because we also have questions of why the queen phase stays as long as it does. Queens can live 5 years as well, and so technically they can live up to 5 years, but they only can lay, I think, like 3 eggs a day. So this species, and like a lot of the sort of ants that have primitive traits, they have really low fecundity. So they're only producing maybe 3 eggs a day. If you have a gamma gate can lay 1 egg a day, so less than a queen. So that's maybe one idea why there's more than one. It's that at least to match the reproductive output of a queen, you have to have 3 gamma gates that are laying 3 eggs a day. But typically they'll have, 8 or 10 gamma gates, and they'll double the reproductive output of a queen. So we see that gamma gate colonies, when you collect them in the field, gamma gate colonies tend to be larger than queen colonies as well.
Okay, so it's colony size, the original colony size, that kind of determines the number of gamma gates. And you said after a tournament, it's relatively stable and there's not much competition. But how do these gamma gates in stable colonies interact? And if you would manipulate them, let's say you cut off 2 legs of half of them, or half of them you put more weight on by giving lots of pain, do you think this would change their social status, or it would be really totally stable, independent, whether you manipulate competitive ability of them or not?
Yeah, there's a graduate student working in my PhD lab now who's been removing gamma gates like one at a time to see at what point that destabilizes the hierarchy. And so what happens is if you have like 8 or 10 gamma gates and you remove one, that usually doesn't do anything. So, and if you ask first, I guess I should say, how they interact with each other. And so once they become gamma gates, they move to the brood pile. They typically stand really tall in the colony, so they stand up high on their legs. They like to stand on top of pupae, so they stand on the pupal cocoons. And so this is how we identify them, because of course they don't look any different from the workers. So if you look at a colony, you can see the ones that they don't move very much, they stand really still. And if they come in contact with another gamma gate, typically they just back up. So they don't engage in those agonistic behaviors anymore. They just retract their antennae and they kind of keep space around them. If a subordinate worker approaches the gamma gate, they actually bow their heads down, kind of like a dog displays submission. They'll put their head down, they'll pull their antennae back, and they'll kind of back away. So that is how you can recognize them in a colony. But yeah, what happens is you can remove like maybe 1 or 2 from the hierarchy, but then eventually you hit a threshold in which the colony itself, for whatever reason, probably they sense that there's not the same level of fertility signal that this pheromone content that's present, something related to that. And instead of just adding new gamma gates, what happens is they overthrow the entire current gamma gates. A new tournament starts. The old gamma gates by that point usually lose. I think a lot of them some of them can hang out. So it's not completely overthrown, but it throws the whole colony into turmoil again. And then they go through a tournament.
Thank you. Lauren, you want me to bring the questions from YouTube, or you first go through the chat?
You can do YouTube. Go ahead, please.
So on YouTube, let me see with us, because it's Mena Davidson is telling, wow, exciting talk and cool system. I'm interested in determination of the castes. If I understand correctly, stress induced on the worker larvae from biting is an important part of the mechanism. Is it possible for larvae to become stressed from outside factors like limited food, predation, parasite pressure, such that a colony develops very few queens, even in the absence of biting behavior? So the question is, I think, whether something else in biting behavior can be the stress inducing caste development.
Yeah, I think that's a good question, and yeah, that makes that kind of speaks to regulation of queen production across all ants at that point, right? If you have a stressful year, then maybe that it would make sense that there's a mechanism that larvae revert back to workers instead of adding extra stress by putting queens into the colony. So yeah, I think stress is probably generally a pathway, whether it's mechanical stress, whether it's nutritional stress, like you might see in other species. I haven't looked at cold stress. It may or may not. In some species, they actually require larvae to go through a winter cold period before they can become queens. So in some cases, the cold just shuts them down enough that they diapause for the winter and then they come back out. So I don't know if temperature stress would be a component, but it easily could be. I wouldn't rule it out. But yeah, I think that's a good point that these nutritional and stress pathways might be interrelated with each other.
Thank you. Then there's another question from Max Verhey. He's one of the students participating to get a certificate. Max is saying, first of all, thank you for a great talk. I'm a master's student from the Netherlands and I'm thinking about doing a PhD. So my question deals with logistics. While the system is really cool, your research does seem very fundamental. So I was wondering, how do you convince grants to fund fundamental research? A very big question for everyone.
Yeah, there you go. That's a good one. So this one, I was fortunate in that when that I started grad school, that was just the time when people got interested in the genomics side. And so my advisor, we already had colonies, so we keep them. To give some background of the scale that it requires to do this research, as a grad student, I maintained around 250 colonies of Harpegnathos in the lab. It took 4 hours to feed them all, and I had to do that twice a week. Thankfully, while I recruited undergrads who would do it 1 day a week, I always still did it one. We went through 10,000 live crickets a week just to keep them fed. So it was a huge operation. Partly it was Bert Holdubler, who was the head of our group at Arizona State. He had funding through his position as like sort of an Uber head of the department to keep some of this going. But then when there was interest from the genomics side, our lab got funding from HHMI, and it was an obscene amount of money for someone like me who does basic research. And so that what supported a lot of my graduate stipend working in that. And so that allowed us, we sequenced the genome at that point, and then and they become increasingly a model for genomic research. And so because one thing that makes them good to be a model is that you can reproduce them in the lab. So the majority of ant species you cannot reproduce in the lab. They require external conditions to trigger a mating flight. People have tried and failed. I think it's just really difficult to get queens, virgin queens to mate in the lab. But Harpegnathos, we can actually, because they mate with their workers, the workers mate with males in the lab, we can actually do crosses. So we can isolate newly closed workers with males from different colonies, and we can observe them mating in the lab. And then because they're totipotent, we can let them start their own colonies. And we can also, if we need more colonies, just divide them in half and wait a month, and then they establish a new hierarchy, and then you have 2 instead of 1. So we've actually distributed our colonies to a bunch of different labs. So there's now a bunch at the NYU Medical School, UPenn. There's a lab at UPenn that works with them, a lab at University of Florida. So they are popping up in more places, and I maintain collaborations with some of those labs to do some of the basic work, because a lot of the big genetic and genomic questions, as I argued in my talk, require this basic understanding, and I think that we benefit a lot from collaboration. I'm more interested in the basic side of it. I'm not a bench scientist, but so I appreciate the opportunities to collaborate with those people who are.
So thank you. These were all the questions from YouTube that are there at the moment.
Thanks, Carsten. So our next question's in the chat. It's from Cheyenne Smith. Cheyenne's a master's student in my lab at the University of Tennessee at Chattanooga. She says her microphone doesn't work right now. So her question was, when the gamma gates revert, is there any difference in the venom? Because you said they revert to biting, but she wonders if the venom was still as effective since they reduced this before as other workers.
Yeah, so I didn't present those data, but we did look at venom. And so the venom gland re-expands also. So it re-expands to the size that it was. They can subdue crickets fine. So that's one of the other plastic components. That's actually was fairly unique. And a lot of ants, they only produce venom during the first, right shortly after they close. And once they use all their venom, they can't produce more. That's true of fire ants, for example. Fire ants, they have a fixed amount of venom and they don't continuously produce venom throughout their life. But Harpegnathos is somewhat unique among ants in that they do continuously produce venom.
Interesting. Next question comes from Clara Jones.
Hi. Great talk from a major transitions approach. I'm Clara B. Jones, study mammals, social evolution. I have a question about workers and whether or not you find age polyethism or temporal division of labor so that the younger ones do the less risky tasks and the older ones do the more risky tasks.
Yeah, that's exactly the case. So as it's pretty typical in ants to have this age-based polyethism where the young workers, when they first close from the pupae, they take care of brood inside of the colony. They don't leave the nest. And then as they get older, then they transition to the more risky tasks. So in Harpegnathos, they switch to moving closer to the nest entrance, and they function as like guards, nest guards. And then the oldest workers are the ones that leave the nest and then do foraging behaviors. What was kind of interesting to us is that for the gamergates, typically younger workers are the ones that win the tournaments, but it's not always. So we've actually taken the oldest workers in colonies and isolated them, and they can become reproductive. They just tend not to win the competitions as much. So typically you have not the youngest workers in the colony, but not the oldest workers, sort of the middle-aged workers become gamergates. What's kind of interesting is that when they revert from gamergate, they completely skip the young worker stage. They go immediately from former gamergate directly to forager. And that's very, very clear and not necessarily what we would have predicted, but it seems like that policing behavior is a hard push. It probably switches their hormones really quickly to that of a forager.
That's great. I have one quick follow-up. Do you consider temporal division of labor or age polymorphism to be a precursor to morphological, for example, division of labor or more complex reproductive division of labor?
Yeah, I think, in a lot of the changes that we see in reproductives are things that are reflected in that age polyethism as well. So I didn't talk about it too much here, but in those last slides that I showed where we included the younger inside workers and the older foragers, the patterns in the brain, for example, we see match with gamergates. So gamergates tend to have their characteristics revert back towards more like a younger worker. So it seems likely that they're tapping into those pre-existing age-based like plastic pathways and then retooling those. And so it's also true that when they first, young workers, when we dissect them, sometimes have eggs, that they produce eggs. And so, there's a huge literature on this, heterochrony and stuff like Tim Linksvaerd worked on and stuff with this. But, they're co-opting probably, and ants and other social insects, they're co-opting a solitary insect life history and then rearranging sort of the timing of those events to create these age-based polyethisms. And then it seems like they're retapping in that and reshuffling it when they evolve back to having these reproductive castes.
Thank you.
Okay, so I was going to ask a question, but I want to go forward in the queue here. And I think the next one is from Vasuda.
Hi, am I audible?
We can hear you, okay?
Okay, great. My name is Vasudha Kulkarni, and I'm an undergraduate at the Indian Institute of Science Education and Research. This is a very interesting talk. Thank you for that. I had a somewhat general question about queenless ants. These queenless ants and their plasticity in behavior and physiology, could you comment on what this means for the evolution of altruism? Does this indicate like a reversal of altruism of sorts, and why do you think that is the case?
Yeah, so that's a good question, and something that I could have talked about. So this reversion to queenlessness and workers that become able to have re-evolved this ability to mate and take on the role of a reproductive in a colony, it happens in primarily in ants that retain a lot of primitive traits. And so they haven't passed this threshold of no return. And so in working with Bert, when I was a graduate student, him and E.O. Wilson published their book, The Superorganism, together. And so we were all excited as graduate students to buy it. And then I bought it and was like, oh yeah, like I'm an insect researcher. I study superorganisms. That's so cool. And then I start reading his book and I'm like, oh wait, I don't study a superorganism. And so Bert Holdauer actually defines the system that I worked on as ants that are eusocial, but they haven't passed the threshold of no return towards a superorganism, which is really when you have a shift from selection on the reproductive output of individuals within a colony to a full shift to where selection is on the colony level reproductive output. And so, yeah, once you get something like a fire ant or a carpenter ant or something where workers have been reduced such that their reproductive output or potential is very, very low, that reduces any chance of direct conflict where they would try to usurp a queen in a colony and lay eggs. But in something like Harpegnathos, they have really low queen worker dimorphism. So what that means is that queens and workers are really similar in size. And like I said, queens can reproduce maybe like 2 or 3 eggs a day where a worker can only make 1, but that's still much closer than you would expect in some of these species that have a higher divergence in queen size versus worker size. So yeah, wherever you see this is in these societies that aren't necessarily true superorganisms, but where there's still a lot of potential for within colony conflict. That's why some of these policing behaviors originally evolved. So in Harpegnathos, we see this as well, that sometimes young individuals will lay eggs in the presence of a gamergate. And when they do that, the eggs usually get eaten so they can smell if the egg isn't right, and then they'll eat those eggs. And then it also changes their own pheromones so that they smell like they're a cheater, and then they get policed and then knocked back down. So in these societies, there is a lot more potential for conflict. And so it seems like in queenless ants, my guess is that it's a mix that there's low dimorphism and that workers still have relatively high reproductive potential. And then there's probably some ecological factors that have selected for them to push it back to a worker reproductive phenotype, though I don't know much about what those would be, but it does, queenlessness has evolved only in these these groups that retain a lot of the ancestral characters.
That's very interesting. Thank you.
So we have another question coming in from YouTube. This is another question from Max, who asked the previous question about funding research. He said, is there a known reason for the very typical mandible shape in this species?
Yeah, they have cool mandibles. It's actually a multi-tool. If you look up a picture of them online, you can see it. I don't think I even had a great one in the talk. But the reason why they have the really long, like their name Harpegnathos literally translates to harp-jawed. So they have like this harp-shaped jaw. The long mandibles are for capturing prey. So some other species too that are independently evolved to be to hunt live prey, and ants also have these long mandibles, and it just gives them, you know, a greater opportunity or a greater margin of error. So if they don't get the strike perfect, they have a bigger space that they could catch a leg or a part of the body. But then why I call it a multi-tool is that at the base of the mandibles, they have these little teeth that stick out. And those are what they use inside the colony. So if they need to pick up an egg, they use that base of the mandibles to pick up the egg. If they need to chew into a cricket, that's what they use. When they're grooming each other, they use those little teeth. And so they kind of have a multi-tool. So the big long mandibles are for catching prey. And then the short ones, really, like if you cut the big part off, they would look like typical ant mandibles, basically. So they would just have those two little small teeth. But yeah, that's how they use them.
We have time for more questions if people want to come through YouTube or in the chat here. In the meantime, I had a question, Clint, and again, I'm like Carsten. I apologize if I didn't catch it.
Oh,
You
no.
know, trying to balance so many things here too. I guess what I was wondering is, if I remember correctly, in the model you had, policing really sort of was the thing that pushed the, well, policing changed the dominance hierarchy to form, correct?
Mhm.
But you also said that policing took quite a bit of time too, if I remember correctly. And to me, that seemed like it could be an opportunity cost. If you're policing one individual, but you're not winning other battles, does that impact who becomes the Gamergate?
Yeah, and so policing is this weird behavior where it's not a dominant individual who performs the policing; it's a subordinate. And so so what we think why policing exists is that it shortens the length of the tournament. So it pulls out these individuals that are still fighting, but are probably going to lose. And so they still have quite long, 30 days is a long time for a bunch of ants to be fighting and not, laying eggs and catching crickets and doing all the other things. But we think policing is there at least still to pull out some of these individuals. They might use that as a colony also to try to optimize, like Carsten was asking, optimize the number of reproductive individuals. So we have every once in a while in lab cases where a colony will have a whole bunch of Gamergates. And when that happens, they typically never fully settle down. And so what we think is that there is some repression of that fertility signal. So if there's a lot of individuals that are reproducing, collectively they have a lower strength fertility signal than if there's a smaller number of really highly fertile individuals. And so what happens if there's too many individuals, they typically, you'll still see some aggression. That's going to be a drain on colony productivity. And oftentimes, like within a few months, it might induce another full cycle again until they get the right number. So policing, yeah, there's not a cost to the individual that performs it directly because in terms of dominance, but it probably does exist. So they're pulling out individuals to reduce the length of time that they're competing, or they're just trying to reduce the overall number that ends up emerging.
Cool, thank you. I'm gonna pass it on to the next person. Clara has another question. So Clara, please.
There we are, I think. Yeah, I'm always interested to ask social insect researchers, sorry, what do you think about the habit in the literature of speaking about primitive eusocial versus advanced eusocial? And in case others don't know what that means, I'm going to say where primitive eusocial would be workers that are all totipotent, whereas the advanced eusocial would be workers that are more or less a sterile caste.
Yeah, and so I still use those terms because in Harpegnathos it allows us to look at, they kind of do both, right? So they have this advanced eusocial trait and that they have fixed castes that develop at the larval stage. But then because they've re-evolved the ability to mate, they also still do these typical behaviors that you would see in like a wasp society, where all the members are totipotent, and then agonistic interactions usually determine who ends up becoming reproductive or not. And so I do think, like, theoretically, I still use that framework to talk about like how we can use Harpegnathos to look at the origins and the transitions of those things and what might have been what might have transitioned. And so we still use the term primitive. I know primitive has, you know, a lot of, you know, social connotations that people are trying to get away from. And even I try to be careful when we talk about traits in ants to say that, you know, initially when I started working on Harpegnathos, Christian Peters, who originally described them, he called them a primitive species, and he did a lot of work on primitive species. And now we know that's not the right term to call them, and they've evolved for a long time themselves. And so we still refer to them as retaining a lot of what we would call primitive traits, or traits that are similar to the ancestral state. But that is a little bit different. I don't know, was your question partly just the use of the term primitive as from the socialist aspect, or was it just the theoretical side of that?
Yeah, I see both in the insect literature. I don't see it in the mammal literature or the vertebrate literature, but frequently in the social insect literature. And I make the distinction myself because I don't think mammals evolved beyond eusocial, quote unquote, mammals evolved beyond the totipotency or primitive state, such as in cooperatively breeding mammals or in the social mole rats. So I'm just always fascinated by that.
Yeah, and that's definitely how we look at it. And it was interesting because I know a lot of the guests here today are mammal researchers. And I was like, yeah, this is the questions that I'm focusing on are what in the social insect world we think about probably most, are like, how do we pass these thresholds from these primitively social species that are totipotent, and then how do you evolve a queen caste that reduces some of that conflict, like intra-group conflict. And then, like I said, so Bert and E.O. Wilson point out again that even within ants, there's another threshold that Harpegnathos hasn't passed. And that one is a little bit more wishy-washy, I would say. It's not as clear of a line of having morphological versus non-morphological casts. But their argument is that you get to a point where the reproductive potential is so reduced in workers that there is no longer conflict within the colony, and that selection focuses fully on colony productivity and much less on maintaining all these policing behaviors and other things that some of these other species like Harpegnathos have to have in order to prevent cheating behaviors and trying to sneak eggs and stuff like that into the brood.
And this is the chat, so I'm going to ask an even another question. Do you consider slave-making species to be more complex or less a regression to less complex?
Yeah, that's an interesting question. I mean, I think of them in the same way as parasites, right? And there's a whole variety of slave-making ant species. So some and social parasites. There's some that have completely lost worker caste, so they sneak into colonies and then they just lay eggs that are new queens and new males, and then those get taken care of. And then there's species that raid the nest of other species and bring and those are sort of the most famous slave-making ants. But that's technically not true slavery because they're working, it's a cross species really exploitation. And then there's other ant species common when I was at Arizona State in the desert, there's honeypot species that actually raid nearby nests of the same species and bring those larvae back. So there's quite a large diversity of how that phenotype or like sort of levels of slave making and different things going on. But you do see within those colonies that, you know, typical in parasite evolution, that they lose certain traits that they simplify because they simply don't need to maintain. Like brood taking, like characters that take care of the brood. But one of the most famous slave making groups is called Polyergus, and they're common throughout North America and I think Europe as well. And they're famous for having sickle-shaped mandibles. Their mandibles literally are these really tiny points, and they are perfect for like stabbing through the skull of another ant. But they're really, really bad if you need to delicately pick up a larva and take care of it, right? And so they're a case where they've specialized on like conducting battles with other ants, but they've probably lost a lot of the behaviors and even some of the physical characteristics needed to do the brood care side of things. So yeah, I don't know. If I wouldn't necessarily say that it's, some of them are just more specialized, I would say, than others.
That's great, thanks.
Are there any other questions that I can relay to Clint from folks? I mean, this has thus far been a great discussion. I'll just check YouTube real quick. And Clint, normally what we do is after the talk, sometimes some of us just stick around and chat if you're able to do that. If not, I just totally understand. You might have other things, and I know you have meetings later today. But just at this point, I want to formally thank Clint for a really cool talk. One thing that I've seen you give 2 talks, Clint. It's always like you're telling a story, which is really enjoyable. So I really enjoyed it, and thanks to everyone for being here.